Hello, my name is Mr. Napoltz, and this is my second lecture on the topic of DNA to proteins. What is the process between DNA and the proteins we see in our cells? Today, we're going over the topic of RNA processing. Last lecture, we began learning how the DNA in our cells contains genes that code for the proteins within and outside our cells, performing a wide variety of jobs throughout the body. We studied the process of transcription in our last video, which is the creation of messenger RNA based on the nucleotide sequence of the gene. To review, we saw how RNA polymerase attaches to the promoter region of the gene, which signals where transcription should start and on which DNA strand it should be done. The RNA polymerase unwinds the DNA and creates a new mRNA molecule on the template strand. This can also be called the non-coding strand, the minus strand, or antisense strand. The template strand is read by the RNA polymerase in the 3' to 5' direction, creating the mRNA molecule in the 5' to 3' direction, matching every nucleotide base of the RNA with its counterpart on the non-coding strand. And it uses uracil instead of thymine in RNA. Now, we have a fully formed mRNA molecule. The RNA polymerase has been notified to stop transcription through the termination sequence on the template strand, but before the mRNA can carry the code to the ribosomes to make the protein, the information on the mRNA is actually faulty. There's coding that does not need to be there. The mRNA needs to be spliced to become mature enough to exit the nucleus. In this video, we're going to go over the key steps of RNA processing. Number 1 is going to be the addition of the poly A tail and a GTP cap. And 2, we're going to go over the excision of introns, splicing, and the retention of exons. And we'll end this video with a discussion on the concept called alternative splicing. First, the addition of the poly A tail and GTP cap. We'll be referring to the pre-processed mRNA as pre-mRNA since it's not mature yet to be translated. The pre-mRNA will have 2 processes done to each end. 1 at each end. Sorry about that. During transcription, once the 5' end of the pre-mRNA is transcribed, a 5' cap is placed on the 5' end. So this is happening during transcription. Remember, the transcription is going from the 5' to the 3' end, which means the 5' end will be the first thing transcribed. Once it is transcribed, while transcription is still going on, the 5' cap is put on. This is also called the G cap or the GTP cap because this cap is made up of a guanosine triphosphate molecule. This has a variety of functions. It, 1, can help bind the mRNA to the ribosome for translation, and it also can be a signaling molecule that keeps ribozymes, which are enzymes that digest RNA, from digesting the mRNA once it exits the nucleus. On the other end, the 3' end of the pre-mRNA, a poly A tail is placed after transcription. Now think about it. Because it's being transcribed in the 5' to 3' direction, the last thing to be transcribed in the pre-mRNA is the 3' end. So it makes sense that the poly A tail will be placed on after transcription. It is composed of anywhere between 100 to 300 adenine nucleotides. Its main functions are to help the mRNA exit the nucleus and stabilize the overall molecule as it travels throughout the cell to the ribosome. The second major step in mRNA processing is the removal of introns and the splicing of exons together. But what do those words even mean? What are introns and exons? Introns are the pieces of mRNA that are non-essential, as in they are unneeded information for the particular protein that is being asked for. Exons, on the other hand, are the pieces of the mRNA that have the needed genetic information for the protein. So we need to be able to take out the non-essential information and keep what is essential for making the protein. So also another way to remember this is that introns need to stay in the nucleus, as in they need to stay inside, introns, while exons need to exit the nucleus. All right, it might be a little reverse thinking, because it may be very easy to think, oh, introns is what stays in the mRNA and exons leave the mRNA. Actually, no, it's more related to where they are located in relation to the nucleus. Introns stay inside the nucleus after they are cut out, and exons leave the nucleus after they are put together and make up the mature mRNA. This whole process of removing introns and splicing exons together is called RNA splicing. So, here's how this happens. Between introns and exons, there are small base pair sequences that are called consensus sequences. And they're called that because there aren't many differences between consensus sequences, as if they have all come to a consensus on a similar base pair sequence. These are important because particular molecules that perform the splicing attach to them. These splicing molecules are called small nuclear ribonucleoprotein particles, or SNRPs for short. Let's use a picture where there are two exons that have a single intron between them to show how this works. The first SNRP on the scene would recognize and attach to the 5' exon-intron boundary. The second SNRP will attach to the 3' boundary between the intron and the second exon. Then the two SNRPs will interact with each other and some other proteins to create a complex called a spliceosome. Once the spliceosome is in place, it will cut between the exon and intron at the 5' boundary. Then the intron will be made into a little lariat, a little loop. But now we have a 3' OH group at the end of the first exon, so it will react with the 5' end of the second exon. During this interaction, the intron is cut from the second exon by the spliceosome, and the two newly freed exons will be spliced together by the spliceosome. Once this has occurred throughout the entire gene, cutting out all the introns and splicing together all the exons, in addition to the 5' cap and the poly A tail on the other end, the mature mRNA exits the nucleus to find a ribosome where its information can be translated into an amino acid sequence that will make up the desired protein. But before we end this video, I want to go over alternative splicing. Alternative splicing states that one gene can be spliced in multiple ways, allowing one gene to code for multiple proteins. And one final side note, prokaryotes don't have RNA processing. The mRNA that is transcribed is exactly what is translated to the protein. But for eukaryotes, for us, alternative splicing during RNA processing allows for various options to choose from for every gene, making DNA coding in eukaryotes much more versatile and complex than prokaryotes.